Okay, well, hello everyone. Thank you so much for joining us today for our presentation in the ongoing COVI lecture series, the 2021 edition. My name is Sarah Ackles, and I'm the Communications and Marketing Officer at Brock's Cool Climate, Enology, and Viticulture Institute. Some of you might know us as COVI. I'm going to be moderating the lecture here today for you. Okay, well, hello everyone. Thank you so much. The lecture is going to run about 45 minutes today, and there's going to be a question and answer period that will follow that. If you can just put your questions into the chat feature at any time during the presentation of this lecture, I'll be sure to share them with the speaker at the very end. So I'm very pleased today to welcome our speaker, who is Dr. Tom Lowery. Tom is a graduate of the University of Guelph and has nearly 25 years of research experience on sustainable grape pest management. That research includes chemical and biological controls, leafhopper anti-feedants, and the use of beneficial vineyard ground cover vegetation. Since 2011, he's also been conducting research on the epidemiology and management of insect-borne plant viruses, including work with grapevine viruses and their vectors. He's currently a research scientist in entomology at the Agriculture and Agri-Food Canada at the Summerland Research and Development Centre out west. He's an associate professor with the UBC Okanagan in Kelowna, and he's also a professional affiliate with us here at COVI. He produced the Insect and Mite chapter of the BC Production Guide for Grapes, as well as accompanying photo guides for grapevine pests and beneficial insects. Tom has also completed research on turnip mosaic virus at Omafra, as well as vegetable IPM research at AAFC here in Vineland, Ontario. Today, Tom is going to give us an outline of the research project, development of sustainable management practices for leafhoppers on grapes. That's being done in conjunction with AAFC and the CGCN. He'll follow that up with an overview of the pest status of leafhoppers and the results of research to develop sustainable methods for their control. So with that, Tom, I'll get you to go ahead and please share your screen for us, and we can go ahead and get started.
There we are. 1 second. Thank you very much, Sarah. It's a pleasure to present today. I'm going to put this on projector screen. Can you just nod, Sarah, if you see that clearly and you can see my cursor and you can hear me? Great.
We're off to all's good.
We're off to a good start. Well, thank you very much. As Sarah mentioned, I'm going to talk about research I've conducted over many years looking at development of a sustainable pest management strategy, particularly for grapes. So before I get into that, I'd like to give you a little bit of a background on my research program, and it's composed of 3 related areas. I work on integrated grape pest management, so looking at all the different pests, and there certainly are enough of them, economically important crop with many and diverse pest and disease problems. So it's important to work on grapes. They have a lot of problems that have to be dealt with, preferably in a sustainable manner. I also work on the epidemiology and management of insect-borne plant diseases. And as background for grapes, there are more viruses recorded from grapes than from any other plant. Grapes are also subject to a number of phytoplasma diseases, and those are vectored by insects that I deal with in my 3rd focus area, which is biology and management of homopteran insects. And these are insects like aphids, leafhoppers, scale, mealybug, and the like. And homopteran insects are important vectors of plant viruses. So aphids are the #1 as far as spreading viruses. But on, for example, phytoplasmas, it's leafhopper-borne are a lot of them. And also grapes are host to many economically important homopteran pests, including several species of leafhopper. So these all come together into a sort of comprehensive program that looks at all these various areas. So I mentioned that I've been doing this work for quite some time, trying to develop a sustainable program for leafhoppers, and it's still ongoing. I just wanted to mention that we're in the middle of a 5-year project. The CGCN and AFC joint program that it's a nationwide, there's lots of projects in it, and one of them is this Leafhopper project. I'm the lead on this project, and I work on the topics that I just mentioned. There are a number of areas that we're working on, and we have great team. Paul Abram works out in the Agassiz station on the Lower Mainland. Pat Bowen, Carl Bogdanoff, and Dave Neild work at the Summerland station. Tara Garapi is in London, and I'll come back to this later why this is important. She does insect molecular taxonomy. Dr. Joel Kitts at Ottawa, he's the taxonomist specialist for Homopteran insects. Justin Renkema is at your AFC Vineland Center. An entomologist, works on tree fruits and grapes. Dr. Sergei Triapitsin is the North American expert on the taxonomy of Anagras wasps, and I'll explain what those are later. They're quite an important component of a sustainable program. So these are the team members, and lots of other people, of course, involved as well, all the technical help. So that's the background, why we're doing, sort of why I'm involved in Grape IPM work. And I want to focus on the cast of characters and give you a bit of a quick run through. So on the top left here, we have two species that are serious pests of grapes in BC. Both of them are invasive species. One of them, the Virginia creeper leafhopper, is invasive from eastern North America. So it's native to Ontario and eastern North America. The western grape leafhopper is native to coastal California and that area, and it's moved north. So these are the two that we have. In Ontario, you have a larger number. You have the Virginia creeper leafhopper. It's native to eastern North America, but you also have all these other ones and more. There have been 21 species recorded from grapes, but these are the most serious pests. So these ones are regularly infesting grapes in Ontario. And you can see some of them are very difficult to tell apart. The western grape leafhopper, the eastern grape leafhopper look very similar. In Ontario, you have three species. These ones here, Erythronura bistrata and Erythronura vitafex, are cryptic species. So they look so much alike, they're really hard to tell unless you put them under a microscope. This one as well, the Grapevine leafhopper, looks quite similar. So you have a large number of leafhoppers in Ontario. I want to mention that photo credits, anything with numbers, came from Andy Hamilton in Ottawa, and they were included in a paper that we published in 2014 on leafhoppers in vineyards in Canada. Other ones, they're credited. If they're not credited, they're one of our photos. So that's the cast of characters. Now, those were the Erythronura pests, and they're the most serious ones, but you do get other ones. The potato leafhopper, Empoasca fabae, feeds from the phloem and injects a toxin causing leaf and shoot distortion and hopper burn. And you can see this isn't even very serious. Sometimes you'll get browning along the edge. The shoots can be distorted and deformed, so they can be quite serious. The previous ones, the Erythronura ones that I showed you, they feed from the mesophyll or the leaf cell. And I'm going to show you damage in a minute. This one is different. The potato leafhopper feeds from the phloem. And because of that and injecting the toxins, the threshold for damage is much lower. So you have a threshold established in Europe for a similar species, Empoasca vitis, and for that one, the threshold is 0.5 nymphs or eggs per leaf. So essentially, if you find any in your vineyard, you're probably going to be spraying for them. The good news is for Ontario and BC, it rarely occurs in BC at all. It moves up each year from the Gulf Coast. So it does not winter in Ontario, so the numbers are variable from year to year, but occasionally you can have significant numbers. We do have a few more. These are technically leafhoppers, but we call them sharpshooters. They're overgrown or oversized species of leafhoppers. And there are a number of those that can cause damage to grapes. The top two, the willow sharpshooter and the poplar sharpshooter, occasionally cause economic damage in BC and have to be controlled. They, as the name suggests, the damage is usually occurring near riparian areas or wetter areas that have willow and poplar. And so they'll build up mainly on those hosts and then spill over into grapes. Another important one that you have in the east is what the Europeans have now named vine leafhopper, Scaphoideus titanus. And it's very important. It does directly feed on grapes, but it has a more important role in that it is a vector of a number of diseases. So in Europe, they had a serious disease called grapevine yellows disease, or flavescence dorée, and it's caused by a phytoplasma. And they had sharpshooters there that would spread the disease. But it turns out that this Scaphoideus titanus from North America is a much more effective vector. And it was accidentally introduced to Europe. So now they have a really terrible problem with grapevine yellows disease. It is also a concern in that it can transmit other phytoplasmas, including and also the bacteria for Pierce's disease. So it's a phloem feeder, important vector of number of diseases. A related group, so a sister group to the leafhoppers, the Cicadellidae, are members of the Membracidae, and these are known as treehoppers. And I'm just quickly going to jump over to this other family because some recent work, some recent concern. And so we haven't normally been too concerned about treehopper damage to grapes. They'll often feed on lateral shoots in midsummer and cause they'll girdle the shoot and cause laterals or secondary shoots to go reddish or even die. And it wasn't a major concern. It happened later in the year. Some new grapes were a concern if a new planting, if they killed the growing tip. That wasn't very good. But a greater concern recently is the possibility that they are vectors of grapevine red blotch virus that some of you may have heard of. So using an artificial diet system, a graduate student of mine, Dieter Kahl, has shown that buffalo treehoppers, and there's a couple of different species of these, all called just buffalo treehoppers, that they are able to transmit grapevine red blotch virus. And a close relative is the three-cornered alfalfa treehopper that is thought to be a vector of grapevine red blotch virus based on research in California. So it sort of supports that idea. The transmission in the field has not been demonstrated yet, but using this artificial diet technique where the treehoppers feed on infected plant material, then they feed into the artificial diet that has various concoctions. It's got sugar in it, but it also has some preservatives, and you're able to then do, excuse me, test that solution for the presence of virus. And the reason for this, why it's able to help determine which ones might be vectors, this type of virus has to circulate through the insect vector. It's taken in, passes through the gut wall into the hemolymph and then back out. So it's a good indicator that at least it has the capability or potential to transmit the virus. So now we're looking at treehoppers differently. They might become a serious pest. What do you do about buffalo treehoppers in Canada? Well, there are no insecticides currently registered specifically for the control of treehoppers on grapes in Canada, but a lot of sprays applied for other insects, including leafhoppers, should provide some control. A bigger concern, a lot of growers are interested in the use of legumes, clovers and things for nitrogen. So they're used as part of a ground cover species mix, and particularly true for organic growers. So unfortunately, these legumes are preferred hosts for the treehoppers, and we've done some work in the field looking at the spread of grapevine red blotch virus, and there's definitely a link with the presence of things like alfalfa or clovers. So I've done a lot of research in the past. These are some photos including looking at different legumes for suitability as ground cover plants in vineyards. And now we're going to have to go back and look at this again because we really need to see which ones of these legumes are the most favorable, which ones support or which ones don't support high numbers of treehoppers if red blotch is present. So getting into the leafhoppers we're really concerned about, these are the Erythronura species that I introduced earlier. So they're give you a bit of background on the biology. So the ones we're talking about are all native to North America, but a lot of them have managed to get around to other places. So some of them have arrived in Europe accidentally. I mentioned the sharpshooter species, but even some of these Erythronura species have managed to get around. I mentioned that the Virginia creeper leafhopper shown here in the bottom left, it's now out in BC in a serious pest. This one originally from California. So they are invasive in a lot of areas, and that's a concern. They are generally all small, around 3 millimeters in length. They produce a lot of young in a season. They have 2 generations per year generally. They all overwinter as adults. The nymphs are often, they're pale, marked with various combinations of browns and reds and oranges and yellows, depending on the species. For most species, eggs are deposited in the leaf mesophyll, so in the leaf layer just under the cuticle into the mesophyll. There are a few that deposit eggs into the veins of leaves. Grapes and closely related plants such as Virginia creeper vine are the only or the preferred host for most of these pest species. A few of them have wide host ranges, but one reason they're such a concern, can cause so much damage, they're specific, a lot of them are specific to grapes or Virginia creeper vine. So that's the biology, and I've outlined a little bit why they're a concern, but they cause also a lot of direct damage to grapevines. So they puncture the leaf mesophyll cells and remove the fluids, which reduces the photosynthetic activity. They reduces carbohydrate accumulation and delays ripening of the fruit. They produce a lot of excrement that fouls the leaves. They also cast a lot of skins along the way. They produce feces and honeydew, and that can culture sooty mold fungus. They're also an annoyance to vineyard workers when you get large populations of these leafhoppers and you're trying to work in the vineyard. They can get up your nose and in your eyes and things. Fortunately, for the Erythronura species, the true leafhoppers, there's no documented field transmission of any grapevine viruses or phytoplasma diseases. So one thing that you have to take into consideration is that the pest status for these leafhoppers differs between regions. So in Ontario, as I mentioned, you have over 7 species. You have 7 serious species, but you have others. They're all native to the region. We've just started the work as part of this 5-year project I mentioned, but appear to have a large Anagras parasitoid populations because the parasitoids are native to that area as well. As I mentioned, the Vitis vinifera and hybrids are grown in Ontario. So there is a split. There's a lot more hybrids that aren't as susceptible to leafhopper damage. You have in Ontario several major grape pests that typically have to be treated every year, and a lower percent organic or sustainable production. You combine all these, leafhoppers in Ontario really haven't been a major concern for the industry, and I've not seen it show up on the priority list to date for the minor use priority list. We're moving over to the west, over to BC. There's only 2 species of the Erythroneural leafhoppers, but they are both invasive pests. And unfortunately, because they're invasive and not native to this area, there are small Anagras populations, particularly, and I'll get into this in a minute, the species of wasp, parasitic egg wasp, that goes after eggs of the Virginia creeper leafhopper. Mostly grow vinifera grapes out here. There are a few major grape pests, so a lot of years, a lot of vineyards don't have to be treated for insect pests, so the leafhoppers are able to escape control. There's a larger percent organic or sustainable production. So leafhoppers, these Erythroneural leafhoppers, the 2 that I mentioned, are both considered major pests out in BC. So what do we do about them? So I've given you a little bit of background. We have to control them. Chemical control is still the main means of control. So leafhoppers are controlled directly with sprays, or indirectly due to insecticides applied for other pests. Now, there's concerns about the use of the currently registered insecticides. For example, resistance of the Western grape leafhopper to DDT was documented in 1953. It was one of the first records of resistance to DDT in an insect. Since then, it's also been documented to become resistant to carbaryl and endosulfan. And I included this picture again of the damage. This was damage caused to grapes at a Penticton site in 1998. And the reason, this is not fall and early frost or anything. This was damage in late August that occurred because the grower, the owner of the vineyard, didn't realize, no one knew that this Western grape leafhopper had arrived in the Okanagan. Excuse me. And the grower had applied 4 sprays of carbaryl to control what he thought was the Virginia creeper leafhopper, which is susceptible. But as you can see from what happened, it simply killed off the Anagras, the little parasitic wasp, and released the Western grape leafhopper from control. And you ended up with this serious, serious damage. There are other concerns regarding insecticide use, the ones you've all heard of: human health, environmental impacts. This picture here on the right shows damage to non-target organisms, including predators and parasites. There's also concern in Ontario and BC with the vineyard housing interface, where spray drift, etc. Concerns of members of the public about sprays that are being put on in the vineyards. We have a general lack of effective organic materials in Canada, and as more of the industry shifts to organic and sustainable, we'd really like to address this concern. So sustainable and organic grape pest management programs focus more on non-chemical approaches, and so I'm going to be presenting a lot of those. But before I do, I'll just close with part of what we're doing now as part of this big 5-year project. We are interested in evaluating new spray materials. So there are a number that we're interested in, but you need to do the research to evaluate the effectiveness, and this can be done through lab efficacy trials or field spray trials. But there are a number of new products we'd like to assess: insect growth regulators, microbial insecticides, horticultural summer oils. We have one, but I've heard they're hoping to get Pure Spray green horticultural oil registered soon. There's also insecticides with new chemistries, and some of these are either more specific against leafhoppers, or they might be better as far as resistance management or softer materials. We face three major impediments when we're trying to register new materials, softer materials in Canada. Wine grapes are considered a minor crop. So they fall into a different category. A lot of times the acreage is relatively small. Companies don't want to invest in all the work required to register a crop, register an insecticide for a crop that's a rather small acreage and wouldn't get used very often. We have to generate efficacy and other data that's not required in the States. And most importantly for the organic growers, we've been unable to have neem-based organic materials registered in Canada. And just a quick mention on neem. It's the most widely used organic material worldwide. It's forms the cornerstone for organic tree fruit and grape production around the world. So unfortunately, we don't have that or a lot of other organic materials. So there's a lot of work to do. So chemical control, moving on to biological control. Leafhoppers have many natural enemies, including European earwigs that scrape eggs from the leaves. So you'll see these patches where the leaves are sort of scraped, the cuticle and the underlying layer where they will eat leafhopper eggs. And they can consume quite a large number of them. But of course, there's a lot of other predators as well. Lacewings and beetles and different things. But the most effective natural control agents are species of Anagras wasps that parasitize leafhopper eggs. And there's a little photo up here on the right. These are very, very small, little tiny wasps. They're also called fairy flies. Some of the smallest insects in the world smallest known insects are these little fairy flies. So they're the most important natural control agents for leafhoppers. So there are various ways to preserve and enhance leafhopper natural enemies, including increased vineyard plant species diversity and the choice of spray materials. So we've been doing over the years a lot of work on that as well. But I want to focus on the most important ones, the Anagras wasps. And it turns out there are several species of these, and they are quite specific. So starting with the Western grape leafhopper, its eggs are parasitized by the species Anagras Erythronura. And so here's a picture of the leafhopper up top right, and the middle picture shows it's actually Anagras danii coming out of a Virginia creeper leafhopper egg, but they look very similar. So these have been, the Western grape leafhopper is controlled very effectively in most vineyards. The exception is some areas where, as shown in the picture on the bottom right, when pesticides disrupt the natural biological control that's occurring. So why this parasite is so effective? It has a large number of alternate hosts. So it utilizes the rose leaf hopper on a number of species, on roses, plums, apple, blackberry, etc., etc. There's a lot of members of the Rosaceae plant family. And we can go and get a plant like a blackberry bush, and we can extract Anagras from that, and we can collect hundreds and hundreds from just a few canes. So it has lots of sites where it overwinters. And also it has some good spring and summer hosts. For example, the mint leafhopper on garden sage, lavender, catmint, etc. is another important host that helps build up numbers. So we have a very effective Anagras parasite that's effectively controlling the Western grape leafhopper in most cases. Switching over to the Virginia creeper leafhopper, and reminder, this is the one that's native to Eastern North America, occurs in Ontario. In BC, we have a different situation. So its eggs are parasitized by Anagras danii, and both of them are native to Eastern North America. But it is mostly ineffective for the control of the Virginia creeper leafhopper, except in a few riparian areas late in the season. And I'll come back and show you an example of this later. So we've been looking for years. People in Washington State have also hunted, and we cannot find the winter hosts. We know the numbers are low, so it has few winter hosts. We don't know what they are. So part of the project we're doing now, we're having another look at this again, but of great importance. They're also looking in Ontario, and I'll get back to that later, but if they can find it where it's wintering in Ontario, that would help us out, that we could maybe find out where it is out here. The idea being we might be able to plant or boost the wintering numbers. Leafhoppers on, there are a few, red osier dogwood looks like it's a summer host. So there's ongoing work on these, and here's a picture in the lower right of the parasites. The first 3 eggs here are parasitized. This one with the opening, and it's just come chewing its way out. The 4th one on the very right side is not parasitized, so it's still sort of an opaque color. And so for this species, you can tell which eggs are parasitized later on. So the work in Ontario, things have improved recently, and I mentioned that Tara Garapi does molecular-based insect taxonomy diagnostics. So in collaboration with taxonomist Sergei Triapitsyn from California and Joel Kitts, who does the leafhopper taxonomy, Tara Garapi is able to utilize molecular diagnostic techniques to directly identify and associate the Anagras wasp species with the species of leafhopper host egg. So we're able to dissect out these parasitized eggs, preserve them, send them to Tara. She can extract from that and run the tests and simultaneously determine the parasite and the leafhopper host. So we do have other work as well where we're just collecting the Anagras and the leafhoppers from, for example, dormant wood, and there's a lot of effort going on to see if we can sort out the various host relationships in Ontario as well as out here in BC. So here's an example of what we're faced with for the Virginia creeper leafhopper and the Anagras danii and why it's not working. Excuse me. So this is a site that actually has good parasitism rates. It's near a riparian area, and there's 2 peaks in the population growth. So here's the first bit. Now this is nymphs. The eggs would occur a little bit earlier. Here's the 1st generation peak in nymphs and the 2nd generation peak in nymphs. And if you look at these bottom lines down here, don't worry about the over and underestimate of the parasitism. The main thing is they pretty well miss the 1st generation entirely. There's really not a lot of parasitism there at all. And then you start getting some later into the year. This is a little bit of an artifact from very few eggs laid later in the season, so the parasitism looks higher. But our big problem is if we were doing this with the Western grape leafhopper, you'd see a big peak early in the season. So that's why they're very effective. So we really need to find out where the Anagras danii is overwintering. So I already mentioned some of these preserving and enhancing parasitism of leafhopper eggs. So how do we enhance these Anagras species? Well, we've done some work to show that increasing vineyard ground cover species diversity helps a bit, not a huge amount, but you get higher parasitism if you have a mix of broadleaf plants, different species in the vineyard. Providing plants for alternate winter, summer, or spring hosts. So again, these plants that host leafhoppers that are used in the winter or the spring. So I mentioned the roses that support the rose leafhopper that's used for the winter, and dwarf cat mint that is used for the mint leafhopper in the summer. And both of these are alternate hosts for Anagras Erythronura that goes after the western grape leafhopper. There's possible transfer, and we're looking for this one. We're possible transfer of a third species of Anagras called Anagras CCOVI from Ontario to BC. So it's native to the east, and it was transferred to California. It's made its way up to southern Washington state, so it's approaching BC, but this one actually goes after both species. It goes after the western grape leafhopper and the Virginia creeper leafhopper. So we might be able to, either will arrive on its own, or we might be able to bring it in. And of course, the most important thing, as I showed by those pictures earlier, is to use softer materials or selective use of insecticides so that you don't disrupt, kill off the Anagras parasites and favor the leafhopper. Switching to the next thing, cultural control. And this one's a area I just wanted to demonstrate the range of work that I've done over the years. This is simply looking at the use of yellow sticky tape that some organic growers utilize for leafhopper control. It's not widely used, and I'll explain in a minute why not. But ran trials where we had large plots, 7 rows, 3 panels, no tape, every row taped, every second row taped, every third row taped. Because another important factor was, or what we were interested in, if you reduce the amount of tape, would the yellow color still attract enough leafhoppers that you could still get very good effective control without having to go through the expense and labor of taping every row? So that's what this was meant to do. We did some other trials as well. So I'm not showing the actual results. I'm just going to show you the reduction. So this was done in 2 vineyards, 4 replicated blocks per site. When you taped every row, you could get a reduction in eggs of 97%, around 90% for the nymphs. There was probably a bit of movement of nymphs from row to row. When you taped every second row, it dropped, not for the eggs, not down to the same level you would expect if there was no attraction or the adults weren't flying from an untaped row to a taped row. So the yellow tape was attracting the adult leafhoppers to adjacent rows, but really not a huge amount when it came to the nymphs. Really weren't getting that much more than what you would expect if you based on the proportion of the rows that were taped. So the conclusion is that the taping itself is very effective. It reduced numbers by 90 to 96%, but the effect is largely limited to the taped row. So you could maybe get away with taping every second row, but it's not going to reduce them to the level you might want. It is very expensive and difficult to apply, so not widely used, and probably only be used in a few hot spots. So if you had leafhopper numbers really low, and often when they winter, they will go into sheltered areas, vegetation outside the vineyard. So you might be able to get away with a row or 2 of tape in hot spots. So not of general use, but some growers do employ that method as well. Another cultural control method that's proven to work quite well and been adopted quite widely is the Canopy Management, excuse me, related to removal of basal leaves in June. And this can be very effective for the control of leafhoppers. It has secondary benefits as well. It also reduces disease incidence, things like bunch rots, and improves fruit quality. There's only minimal impact on vine growth or yield, really very, very little, and you could probably adjust for that. So we did a multi-year trial involving 4 treatments applied to 2 cultivars at each of 2 vineyards and showed an average of 70% reduction in leafhopper numbers with removal of these basal leaves. You can see the picture over here. We went up to the second cluster, second fruit cluster, and in one site took all the leaves off and did this by hand and timed it for when just about 5% of the eggs had hatched. So it's fairly effective. Growers, of course, a lot of them, they're doing machine removal of basal leaves. It won't be quite as effective, but it still works quite well. Just wanted to show you graphically what we did. So here's the results from one site, 1 year. Pinot Noir, Chardonnay grapes. Here's the not stripped. Here's the late strip. So a lot of growers used to, and some still do, they will strip leaves starting sometime in maybe August. And there's two different types of early leaf removal that we tried. One was early with a single pass, or 3 passes. They're traditional, where you'd go through and you'd sucker and you'd shoot thin, etc., and then do the leaf removal. So really no difference between those, but you can see quite effective, about 70% control of leafhoppers. So moving on to cultural control, managing vine vigor. A lot of this work has come out of California, where you can control vine vigor with nutrients, but the work I'm showing here was looking at deficit irrigation. So 25 or 50% reduction between berry set and veraison. So it reduced populations of Western grape leafhopper and variegated grape leafhopper. And when they looked into this, they found out it reduced the egg laying by the females. So they laid fewer eggs on the vines that had been exposed to the deficit irrigation. It increased nymphal mortality, and the reduction in number of nymphs ranged from 30 to 54%. And I've got the paper cited down at the bottom here. So that's another method that you can, some moderate deficit, regulated deficit irrigation at the right time can reduce leafhoppers by a significant amount as well. The flip side of this, leafhoppers love vigorous canopies, thick canopies with lots of nutrients. So it makes sense that to go the other way, you can help control them a little bit. I've done a lot of work on leafhopper feeding and oviposition deterrence. So just to remind you, these leafhoppers, the serious pests, are all quite host specific. So they like grapes, they like Virginia creeper vine, and most of them do not lay eggs. The eggs do not survive on other plants. So they're actually inside the mesophyll. They're in the leaf. The leaf is nurturing in a way, if you think of it that way. And even for grapes, sometimes the eggs don't survive, or don't survive well on certain cultivars or certain situations. So they have to be very choosy. And so this work came about because of some situations where there were good numbers of leafhoppers and they simply disappeared. And contacted by growers, in one case we were setting up an efficacy spray trial, had everything all set up, flagging and everything, and they disappeared. And the grower assured us that no insecticide had been applied, but they were gone. So when we looked into the records, it gave us a clue where we might want to look. So we've done a lot of work over the years. This goes back a long way, looking at different fungicides, surfactants, plant essential oils, formulated plant essential oils, and some of this is included in the current project. What we found is that several organosilicone surfactants and strobilurin fungicides are highly deterrent to leafhoppers. And I just showed you one example down here where we did lab bioassays. And it's a very simple procedure that we start with. We eventually go to the field, but simple leaf disc choice test bioassay. We have opposite sets, two that are treated only with the control treatment that consists just of a surfactant, not one that's deterrent, and it took a bit of work to get the right ones. So the other two are treated with a concentration of the test material. And we run different concentrations, repeat this over and over again until we get sufficient numbers, come back in 24 hours, see where they're located. And here's just one example for one trial, organosilicone surfactant called Widespread Max. And I'm not plugging any particular materials. I just pulled this one out to show you. And here what we have is on the treatment discs at these various concentrations. Here's the highest or the recommended field rate, and going all the way down, series of dilutions, and compared to the numbers that were on the opposite pair that didn't receive the treatment. And you can see at the recommended field rate, there was only a small number, 5 of them, of the total 127 that were on the ones treated with the Widespread Max. So this, we set up these tables. We do a series of 3-step statistical analysis on all this work. And this is our first step where we do contingency table analysis just to start things. And then we're trying to come up with equivalent to like LD50 rates and everything. Here's a field spray. So we do the work in the lab, and it suggests which materials would be effective. But you really want to take things to the field and verify that. So this is results from a trial looking at the numbers of Virginia creeper leafhopper nymphs per leaf for plots that were sprayed with field rates of the surfactant of another surfactant, Silgard, and again it's another strobilurin one. Organosilicone product, or the strobilurin fungicides Flint and Pristine. And so looking at the results, you can see a dramatic difference. The spray was put on, and I should have marked this on here, I believe it was August the 4th. August 27th, you can see quite a dramatic reduction in numbers on the vines that were treated with Silgard or Pristine, not so much with Flint. But by September 3rd, Flint was pretty good, but there was greater than a 95% reduction in numbers for the plants that had been treated with Pristine. So it was very effective in the lab and very effective in the field. So it's nice to have that linkage. You can screen things in the lab initially. So again, I mentioned what sort of clued us to look at this. So certain neonicotinoid insecticides are also repellent to adult leafhoppers. And we found out, oops, sorry, I jumped down a page here. So so what happened with this? Neonicotinoids were registered, and they said, these are great, they're systemic, they persist, it doesn't matter when you apply them. What would happen, we try to encourage spraying, doing monitoring, only spraying the areas or the blocks that require it. We very soon receive feedback from industry members who were monitoring, spraying one block, and then finding that the leafhopper, the adults were flying, moving to adjacent blocks. And we'd done a lot of work previously showing that once leafhoppers move into a block, they generally kind of stay there, or they used to. But then we found that, okay, this isn't happening anymore. We're seeing leafhoppers moving not only to unsprayed blocks, but even to adjacent vineyards. And this was a real problem for organic vineyards where you'd suddenly have huge numbers of leafhoppers coming in. Excuse me. So, coincidentally, too, though, that these neonics were started to come on the market around the same time that the surfactants were, the organosilicone surfactants were coming in. So jumping down to the bottom line here, organosilicone surfactants, some of which are labeled organic, became widely used around the time that the new strobilurin fungicides and neonicotinoids came on the market. So it takes some extra work to sort out for these fungicides that are highly repellent and these neonicotinoids. Is it the active ingredient, or is it more likely that it's the organosilicone surfactants? I should mention a product more recently coming on the market, Grandivo. It's a fungicide with the organic. It's a biofungicide, microbial fungicide, if you will. It's also registered in the States for leafhoppers and mites, I believe, as well. But it reportedly also has some repellent action. And it might be that repellent action that's actually giving the effect on leafhoppers. And again, I don't know how that's formulated. A bit of a side story here. When I contacted the makers of Pristine, I told them about the work, and I was hoping to get a blank formulation so we could actually find out if it was the surfactant, the adjuvants, the component, the non-active components, or was it the active material. They were very quick to they weren't interested, and they didn't supply me with the blank, the material without the active ingredient in it. So I suspect maybe they knew about this. So I'd like to summarize all this. So I've given a background on the leafhoppers, their biology, and their importance to grapes in BC. They are becoming increasingly important in Ontario as more growers are pursuing organic or sustainable practices. It is a bigger problem down towards Kingston Way, where you don't have some of the major pests that you have in the Niagara region. So it is a concern in Ontario as well. So there's a desire for spray materials with different modes of action and in the development of non-chemical controls. And the effectiveness of individual non-chemical control practices has been demonstrated. So I've listed a number of them. If you started layering these together, where some things are 95%, some are 70%, some are 50-some%, you could see that the possibilities are there to use them in alone or in combination. And of course, we'll still always use selective insecticides and try to preserve or enhance Anagras parasitoids and other predators in a sustainable leafhopper management program. So a leafhopper management program suitable for other regions does require additional research, but many of the practices that I've outlined can be transferred or adapted for other regions. For example, early season leaf removal probably has a greater benefit in Ontario, where you have higher humidity and you'd really like to, more rainfall through the summer. You might want to open up that canopy a bit more to prevent things such as bunch rots, etc. So a lot of these, I believe, can be easily transferred. I think some of them are relevant already. The Anagras danii is, I believe, very active in Ontario for the simple reason that Virginia creeper leafhopper, while it is a serious pest in BC and reaches tremendous numbers, in Ontario, it's very rare. Of those ones I showed, those 7 species, it's probably the least common. So something is controlling it effectively in Ontario. Here's another way to summarize everything I presented. Here's sort of a layering, if you will, of actions that can be taken throughout the year, starting with planting of beneficial plant species. I mentioned the roses, and some of these can be planted outside the vineyard, winter hosts, or possibly even spring or summer hosts at the adjacent adjacent to the vineyard or perhaps even in as part of the ground cover mix. You might want to use sticky tape, at least maybe in limited amounts on the edges. Moving on from that, you might want to try an oviposition deterrent early in the season before the leafhoppers have begun to lay eggs. A bit later on, when the first nymphs are hatching, you might want to go through and do the basal leaf removal. Or if you're not removing those leaves, you might want to put on a feeding deterrent for when those nymphs hatch, try to drive them off the grapes. Foliar oil, this would be the time that you'd put on oil or insecticide a little bit after the application of these feeding deterrents. So when most of the nymphs have hatched, you might want to try these foliar sprays. This is all for the first generation. Moving into the second generation, you cycle through again a possible oviposition deterrent, a feeding deterrent, and then finishing up with possibly a second oil spray. What would really be useful, one thing I am working on a bit, and hopefully we can come up with a good model for this, coming up with a model of when growers should apply these various strategies. So models based on temperature, similar to day degree accumulation, or plant developmental stages could be used and would assist growers with timing of the various control actions. And with that, I'd like to acknowledge lots of help provided by, first of all, funding provided by Agriculture and Agri-Food Canada and the CGCN through funding partnerships. The BC Wine Grape Council has been very generous over the years. Thank you to a number of technicians, Andrea Bronner, Naomi Delury, Cheryl Zarowski, and Linda Jensen for technical assistance. And of course, numerous students helping along the way. Special thanks to a number of individual growers and wineries for their assistance. And thank you very much.
Well, great. Thanks so much for that, Tom. That was a lot of really great information for all of us there today. We all appreciate it. We're going to switch over to the question and answer portion of our talk now. So we've got a couple questions in the queue ready to go for you now. So I'll start off with the first one if you're all ready there.
Sure.
So the first question is, for timing June leaf removal to reduce leafhopper population, can egg hatch be related to timing of bloom? So example, before, during, or after bloom?
Yes, it can. And the good indicator of that, it's around petal fall or a little bit beyond that. But that's one of the things that I mentioned at the end, where we're trying to come up with these indicators. And growers they're very fond of these developmental stages. You know, you see pictures of when you have the woolly bud stage, you put this on, and so we have to fine tune it a little bit more. There is a bit of a problem. Not a huge problem, but the grapes do develop a little bit. They're not uniform in their development. Some flush out earlier, some a little bit later. But fortunately, the leafhoppers wait generally until the first leaves are fully formed, fully mature, and there's a sufficient amount of wax cuticle before they start laying the eggs. So that brings it back mostly in line with the developmental stage of that particular cultivar. But yes, you can. It's based on very late into just after petal fall.
Okay, great. Thank you. The next question here, would moving row middles affect beneficials? And it's kind of a two-parter, and that says, how far into the vineyard would beneficials move from border plantings of alternative hosts?
So that's a really good question. And one of the main strategies, or one of the strategies for preserving beneficial insects, is to either enhance the drive row vegetation by putting in a mixture of plants. And some people do combinations with flowering plants, broadleaf plants as well as grasses. And that is beneficial. We've done a lot of work not just for leafhoppers, but also for cutworm and other grape pests, that having more species diversity in the drive row reduces numbers of pests, reduces pest populations, and it builds up numbers of beneficials. So if you have a very well-established mixture in your drive row, one thing you can do that's fairly simple, and some growers do it out here, don't mow the entire block or vineyard on the same day, because can quite seriously impact. So what some growers do, taking that advice, they'll go through and they'll mow every second row or every third row, and then come back in a week or so and mow the alternate ones. So that's one way you can do it. You're asking about the alternate hosts on the outside of the vineyards. You really need a good number of them. If you think about the leafhoppers coming out of the vineyards, the Anagras coming out of the vineyards, looking for the alternate hosts for the winter, if you only have a bush or two, they're going to overwhelm that, and they might take out all the alternate hosts that you really want. Good news is that the parasites rarely parasitize every egg. Even when we put them into a cage, lots of Anagras into a cage with a limited number of eggs, they never seem to parasitize them all. So even a plant quite close to the vineyard. So if you have a hedgerow, or if you have a waste area, you could potentially put in plants that would be beneficial. One of the things I like to say out here for the Western grape leafhopper, it's great that the mint leafhopper is a host, because it's on things that are very attractive that you'd love to have around a winery anyway. So you might want to have lavender and different things like that around a winery. So you could put in some of these hardy bush roses or different things like that. I don't know if I answered the question properly. Did that cover it?
Well, actually, my mistake there. Wendy had asked about would mowing row middles, and I had said would moving row middles, but I think you touched upon mowing in your answer. But Wendy can just type in the chat there if you needed more clarification, or if that didn't address what you had asked. So that's my fault, looking at it quickly out of the corner of my eye there.
Okay. I would like to add one more thing, though, is that the movement of beneficials back in after a spray's been applied. And we've done some work on this. First thing to come back in is typically spiders. They move in pretty quickly. A lot of spiders are airborne and they float in, or they seem quite tough. So they get it back in quickly. Some things are much slower. So if you're a beetle crawling around on the ground, it's going to take you a little bit longer to get it back in there. But some places, it can be, if you have a large expanse, and we have some of those, for example, down in the sandy black sage area where there's hundreds and hundreds of acres all put together. If it all gets sprayed out, it can take a while for the beneficials to move back in. So it all depends on the location of the vineyard or the vineyard block, and proximity to the areas that would maintain healthy populations of beneficials. The Anagras do not fly. They're tiny little things. They blow around a little bit, but they don't fly that well. So we see the movement coming in from the edge of the vineyards, and they move quite slowly into a vineyard.
Okay. The next question is about insecticides, and the question is, which insecticides are the hardest on beneficial insects?
Well, you have to look at short-term and long-term. So there are a lot of these are listed in guides. So our BC puts out a list of chemical use for grapes, and they put cautionary notes. I think Ontario does the same thing. So, excuse me. So they'll list that they induce mite outbreaks or whatever. So if you look at the situation with, say, a pyrethroid, they are quite harsh on beneficials, but they don't persist as long as some other ones. So they're broad spectrum. quite toxic, so they'll knock everything down very quickly, but then disappear after a short while. If you look at the neonicotinoids, and a lot of people have heard about the problem with bees, etc., they are quite harsh, and I have to be cautious here. They are trying to change them and come up with slightly different formulations, but I have done work on this. They are quite harsh on beneficials, and they persist. Some of the earlier ones we worked with, you would get an effect carrying into the third year. So you apply it 1 year, a strong effect the next year, and often still a carryover into the third year. So they are quite harsh. You're not going to get a lot of beneficials. Now, the problem with that is that some of our pest insects are less susceptible to neonics than the beneficial ones are. So there can be a tipping point where maybe in the third year, and this happened more with some of the older materials, you might get the pests bouncing back up, for example, scale and mealybug, because there was still a negative effect on the beneficials, the parasitoids in particular.
Okay, perfect. The next question is related to trap crops and whether they could be used for leafhopper management.
Unfortunately, no. Well, let me break it down into two different groups. The Erythronura that I talked about. So the Erythronura leafhoppers, those species that are, and the ones I showed you, they are very host specific. So they want to, some of them only use grapes and related, closely related species like Virginia creeper vine. Other ones, a little wider host range, but still grapes are the preferred, the main host. It would be, you would not be able to use a trap crop for those because grapes are the far and wide the preferred host. So it's not going to work on those. It might work on the potato leafhopper because it does prefer, it likes grapes, but it has a very wide host range. And mainly on plants, preferred host are the solanaceous plants. So it's called the potato leafhopper because it causes feeds on potato and related plants and causes a lot of damage, including all this hopper burn. But it's pretty broad, so I'm not sure on balance what you'd have to do with that and which plants you might have. There are native solanaceous plants in the Gulf Coast area that it develops on, and so that might work. But also there's some types of beans and things that it really likes. So there's a possibility you might be able to sow a plant in the drive row in the cover crop and use it as a trap crop for that species. But I'm sorry, I don't think it would work for the others.
Okay, thanks for that. The next question asks, would having alfalfa cover crops promote potato leafhopper?
The potato leafhopper will feed on alfalfa. It feeds on a wide range. So the potato leafhopper pops up everywhere. It's a very broad host range. So I'm not sure, though, if there'd be much of a difference between alfalfa and some other plants that you would have. You'd have to look at the mixture that would be in there anyway. But I think alfalfa would promote potato leafhopper more than some other plants that it wouldn't like as much.
Okay. That looks like, I think, all of our questions for now. Unless anyone has any others, you can put them into the chat there. But if not, we'll just kind of plug away here. So just wanted to thank you again, Dr. Lowery, for your time today. Really appreciate you sharing this research with us. For everybody listening out there, thank you as well for contributing your questions. It made to a lively discussion, which is fantastic. This video will be posted to our COVI website, usually within a week or so, and that website is brocu.ca/COVI. You can check there for this video in about a week's time. And next week, we'll be back with another lecture. That's March 3rd at 3 p.m. Our speaker will be our very own Belinda Kemp, who's going to be discussing her research on why Ontario wine consumers buy Ontario wine. So another great lecture to look forward to. We hope you can join us then. So again, thank you so much, Dr. Lowery. Thank you, everybody at home. And we hope to see you next week. Have a great afternoon.
Thank you.